Hey everybody, welcome back to Bio229 Microbiology at HCC. I am your host, Mary Annarella, and we are finishing up this last little bit of Chapter 5, which is control of microbial growth. And the goal of controlling microbial growth depends. We might want to kill all bacteria, or we might just want to keep them from growing, meaning keep them from multiplying and making more of themselves, or sometimes, as with hand washing, we just want to remove the bacteria and send it someplace else, like down the drain. And we do this using physical means, such as hand washing, with heat, with pH, with salt or sugar for water availability. Let's see. And so now just this last little bit, we can do it with chemical means. Now just to point out a couple of things here, that chemical means we generally refer to, we use chemicals as disinfectants a lot. So most chemicals are going to be disinfectants. Some are antiseptics that we use on skin. Use on the body. But in the body, we use antibiotics. So while those antibiotics fall under chemical means, we're not going to consider them right now. We're just going to be considering disinfectants and antiseptics, disinfectants that you use like on a surface, or antiseptics that you would use on your skin. So antibiotics taken internally, that's a whole different ball game. So just to talk about some of the different classes, you have alcohols. Okay, first up, we're using 70% isopropanol, like crazy. That's what hand sanitizer has to be. And actually 70%, it needs to be more than 50% to destroy bacterial cells, and generally up to 90%, but 70% ethanol is actually better because you need a little bit of water in there to allow the alcohol to penetrate a bacterial cell wall and actually coagulate all the proteins there. But alcohols will destroy most viruses, and most of them, not all of them, if they don't have an envelope. Sometimes it doesn't work. Let me just do a nice screenshot here. I said yellow. There we go. Okay, so this will destroy most bacteria. Let's go with 70%. Will destroy most bacteria and viruses. Doesn't work against endospores. Not for destroying endospores. So if you still have endospores under your fingernails, which is entirely possible, when we swab people's hands in laboratory, we occasionally do find some endospores there. Alcohol's not going to kill that. But this is a major form of, alcohols actually work as a disinfectant and as an antiseptic. We use it on our hands. So I would categorize those as both. Okay, these aldehydes. Oh my gosh, this is not stuff that you use on the human body or in your home. Think of formaldehyde. And, formaldehyde is a chemical that they use in the embalming process. So it will destroy tissue. And it stinks to high heaven. Oh my gosh, those fumes are awful. Formalin. Am I spelling that right? How do I spell Formalin. Formalin. Yes, formalin. Formalin is used similar to formaldehyde to preserve biological specimens. For example, if we're going to cut sections and put them on slides for pathology. So there is that. These biguanides, and by the way, you're not going to need to memorize biguanides, and aldehydes and tell me what formalin is used for, but biguanides are commonly used in a lot of antiseptics. They put it in lotions. Stuff like, I want to say betadine is used, they use that on catheters, I'm pretty sure. Betadine, what is it? Chlorhexidine. Okay, halogens. Oh, iodine. Actually, betadine has iodine in it. Iodine. Iodine is an antimicrobial, and we use that on skin, like those iodine wipes or those iodine-like swabs that they put on you, and it's a brown color before maybe you get a shot or before maybe someone inserts an IV. They use that. Chlorine, well, think of that as bleach. Chlorine bleach destroys bacterial cells and viruses really, really well. Metal compounds. Okay, Ag, this is silver. And AgNO3, I don't know what the ions are. I don't know if that's like a 2 and that's a 3 there or something like that. But AgNO3 is is silver nitrate. Silver nitrate. Silver nitrate is added in droplet form into newborn infants' eyes. Yeah. And what it does is prevent eye infections from the microbe that causes gonorrhea. It's easier and cheaper just to put these silver nitrate drops in every newborn infant's eyes. It's cheaper to do that than to test every single pregnant woman for gonorrhea. If a woman does not have any symptoms of gonorrhea, why test them? But they might be positive for it, and it's just easier to just across the board put these silver nitrate droplets in all infants' eyes, and it stops infection from, it's Neisseria. Oh, how do I spell Neisseria? Neisseria, oh gosh, don't ask me to spell gonorrhea. Gonorrhea, something like that. It stops infection from Neisseria, gonorrhea. So yeah, that's an important one using this metal compound. I mean, you need to know some of these, but I'm not going to ask you about chlorhexidine, but I will on the exam ask you about silver nitrate drops, about bleach, about alcohols. Phenolic compounds. Ugh, I hate these things. Straight up phenol is the smelliest stuff. It smells a little like formaldehyde to me. But they're fairly toxic. You do not want these around. And one type of phenolic compound that is really familiar is triclosan. It's microban. It was really, really common in, oh gosh, just about everything. God, they put it in antibacterial soaps. So this was in antibacterial soaps. It's still in toothpaste, but I think it was just 2 years ago that triclosan is banned. It is banned now in everything but toothpaste, and why it's allowed in toothpaste, I do, and nothing else, I do not know. But they used to put it in everything. They put it in mouse pads. They put it in lotions, shampoos, like the whole idea, but especially with antibacterial soaps, washing your hands is a means for removing bacteria. And so adding antibacterial substances to that when it's just going down the drain, study after study found that just regular hand washing with plain soap was just as effective as washing your hands with these antibacterial soaps containing triclosan. And the deal with triclosan is that it was found to be toxic to fish, toxic to aquatic life. There was also evidence that showed that when microbes are exposed to this triclosan, and I just have the trade names here, microban and ergosan, that they actually internalized the substance and had the potential for developing resistance to it, in which case it was just going to be rendered completely ineffective. So triclosan finally, after many years, scientists finally convinced the FDA and provided enough evidence for the FDA to ban it. Johnson & Johnson was the first company to use it, and it's still left in toothpaste. So I don't know why it's not okay in anything else, but we're allowed to have it in toothpaste. But I don't know. I just avoid the stuff. I'd rather just brush my teeth and spit the stuff out. There's also evidence showing that triclosan exposed to sunlight in groundwater, when it got into the water system, actually converted to a type of dioxin, which is very, very toxic to aquatic life and to people. So we don't use that stuff anymore. And I have up here, antibiotics, no substitute for infection prevention and control. So we will talk about antibiotic and antibiotic resistance a little later on. There is one more form of chemical that I did want to talk about, but I don't have it on a slide here, so I'll talk about it here. And these are ammonium compounds. And they're very common in hand sanitizers. There are two types of hand sanitizers. Basically. Alcohols, which we already talked about, alcohols, and these ammonium ones, ammonium compounds. And the biggest one for that is benzalkonium chloride. Is it benzyl? Benzalkonium chloride. It's very common in hand sanitizers, and it appears to be very non-toxic. So it's in some hand sanitizers, but apparently it works better as a disinfectant. So for hand sanitizers, we generally use alcohols. Alcohol is the compound of choice there. But benzalkonium chloride is a common one, and you'll probably come across it. It's used in some soaps, detergents, and the like. And that pretty much wraps up Chapter 5 for us. So we are moving on to Chapter 6, which is one of my favorites. We need a different color here. Let's go with a bright blue. There we go. Moving on to Chapter 6, which is metabolism. Ooh, you're in for a great ride here because this is one of my favorites. Metabolism is basically the sum total of all chemical reactions in the cell. Chemical reactions, I am abbreviating reactions, in the cell. Basically, it has to do with how cells make and use energy. And it takes a couple forms. Let's go to Dropbox. Oops. it starts off with catabolism. Now, these are processes that make energy by breaking down food substances to make some ATP. Oops, I'm switching up my little desk here because my neck is bugging me. All right, this is not working. All right, so catabolism is all about breaking down substance to get energy out of them. Our cells break down sugars so that we can get energy out of the sugars that we eat. So this is all about making ATP. You get some waste products from that. Now the opposite of catabolism, getting energy out of the foods we eat, breaking molecules down to get ATP and energy out of them. The opposite of that is anabolism. This uses ATP to make substances that your cells need. Okay, now I finally got this little desk right here. All right, so catabolism breaks down substances and gets energy out of them. Anabolism uses that energy from catabolism to build molecules, because it takes energy to build molecules. There we go. They happen at the same time. Your cells, microbial cells, are breaking stuff down at the same time that they're building stuff. If we waited to break down every little bit of sugar we had and then built everything all at once, it just does not work that way. Cells are continually breaking down substances for energy and then building the molecules that they need. All right, you got a couple of goals of metabolism then. To harvest energy, that's catabolism. Make stuff for the cell, that's anabolism. But in the process of both of these, you get some molecules that we call precursor metabolites. Do this for you right here. Precursor metabolites are these intermediate substances, like let's say glucose is being broken down. Before that whole breakdown process is done, there are a couple of intermediate molecules that can be siphoned off and used in some other pathway for building some other molecule, for example. Like, okay, so the term metabolite, it's just the end product of a metabolic reaction. Let's take a look at what that would look like on the next slide. So for example, if you have this metabolic pathway, and let's snap a shot of that too. Here we go. You'll have this starting compound here, and it undergoes a chemical reaction. That's what this arrow is for. All these arrows mean a chemical rearrangement occurs. And let's say, this is some starting compound with, 5 carbons on it. That's what these little balls will be. I'm not going to draw a whole compound. But, maybe it gets rearranged and it's a 4-carbon molecule, so it's something else. And maybe here it gets rearranged and it's only a 3-carbon molecule. Well, sometimes, one more time, and then at the end, maybe it's just a 2-carbon molecule. Sometimes what happens is that this molecule here is needed for a whole other pathway. And so it gets taken out. It kind of stops this process. This would be called an intermediate, precursor metabolite, if it gets siphoned out and brought into some other biochemical pathway. All right, so back to our little, I'll stay with red. Okay, let's say we've got a starting compound here, and maybe some chemical rearrangement occurs, 3, 4, 5. All right, when glucose is broken down, it's actually broken in half. And what if this thing is just broken in half here? We call that a branched pathway, because it, this molecule here got basically broken into two parts. So you'll get two end products here. And this should look a little familiar here too. When you have a starting compound that combines with something else and they form one bigger one, and then undergoes another chemical rearrangement, and then another chemical reaction, and then another one, and one of the end products here is actually one of the starting ones. It feeds back into itself. This is cyclical. So like the citric, an example is the citric acid cycle. Citric acid cycle is cyclical. Oh, and before I do that, this branched one up here, an example of a branched pathway is glycolysis. Glycolysis is a branched pathway. All right. So, okay. Before we get into the metabolic cast of characters, what this chapter is all about is how, the main portion of it is how cells make energy from foodstuffs. Every cell out there uses carbs as foodstuff. And, even if you eat complex carbs, it's going to be broken down into glucose eventually. So, for example, it was in E. coli that we first figured out and learned about aerobic cellular respiration. And the overall chemical equation, it doesn't happen all at once, but the overall chemical reaction for aerobic cellular respiration is C6H12O6, and this is glucose, combines eventually with 6 oxygen molecules to make 6 CO2 and 6 water molecules, and oh yeah, plus ATP. This is the topic of 90% of chapter 6 for us. This was first described in E. coli. We know your cells do it, and any cells that are aerobic, that are strict aerobes, do aerobic cellular respiration. So just the term aerobic means with oxygen. This makes it aerobic. There we go. So, but the thing is that this does not happen all at once. This looks a lot more like several of these metabolic pathways here. So what's really happening here, this does not happen all at once. Sure, we could go over, well, not now, but in the past and hopefully in the future, we could go over to one of the chemistry labs over in Frost, and we could get a dish and put some purified glucose in that dish, and we could light a fire under it, with a Bunsen burner. Right there. And we could get it to combine with some of the oxygen in the air, and we get some CO2 for sure, and we get some water. We'd probably also get some elemental carbon. It'd be all blackened in the dish. And we would get some energy, but we wouldn't get ATP out of it. We'd just get some heat. And the cell can't use heat for energy. The cell needs ATP. So what the cell does is instead it does this whole reaction by breaking down glucose little by little. And it becomes, a different molecule. One chemical reaction occurs and this glucose looks a little different. And then another chemical reaction occurs and that molecule looks a little different. And then it's split into 2-3 carbon molecules. It's going to be C3H something, O something. I'm just going to put C3 here for right now. And then each of these undergoes several chemical reactions, becomes slightly different. And then eventually what's left of this will combine with oxygen at the end. So by breaking it down step by step, the cell gets ATP out of this instead of heat. So take home message. This doesn't happen all at once. Okay, it happens step by step. Happens in, let's say, in a series of chemical reactions so that the cell can get ATP out of this. Now, these chemical reactions have some players in them. You need enzymes, for example. Some of these chemical reactions are oxidation and reduction reactions. These things called electron carriers are involved. So before we get into the nuts and bolts of how this happens step by step, and again, I know that you have all learned about cellular respiration beforehand, but you're going to be expected to delve more into it. You're expected to have a greater understanding of it in this course, since this is an upper level class. All right, but before we delve into that, I want to talk about what all those little players are. So the players are enzymes. We got to talk a little bit about ATP. We got to talk about energy sources and electron carriers. So first up, we're going to talk about enzymes. This is all review for you, but I feel the need to review it. Enzymes are proteins that catalyze. Catalyze means to speed up chemical reactions in the cell without becoming altered or consumed or changed in any way. They go in the same and they come out the same. They are catalysts. I like yellow on this blue here, blue background. Okay, without, that's what that W/O means, without becoming altered or consumed or changed in any way. They go in the same and they come out the same. I want to, okay, lowers the energy of activation. We'll talk about that in just a sec. If we have a generic chemical reaction, I would say that substance A chemically reacts with substance B to form products C and D. This is just a generic sort of reaction I'm just making up here. Okay, just a reminder that these are called the reactants, and C and D over here on the right would be called the products. If an enzyme works on this chemical reaction, the name of the enzyme is usually or often written above the reaction arrow in teeny tiny letters. So I'll just write enzyme Q here. It'll be written like that. Let's see. Yeah. And let's just go back to, oh, no, it won't let me do that if it's a screenshot. Where's my notability? Here we are. Okay, I just want to say that enzymes are named after the substance they work on. So something like lactose, and you know that's a sugar because it ends in ose, is broken down by an enzyme called lactase. That's what's in those Lactaid pills. For people who are lactose intolerant, they lack the enzyme lactase. If you have a puppy, like I do, although she's technically not a puppy anymore, she's an older dog. Dogs need to be house trained, and so you buy those little puppy pads and train them to pee on those, because if they pee on the rug, you know that's going to smell. And if a dog pees on the rug, and oh gosh, cat pee is even worse, it really smells badly. That's because there's a substance called urea in urine. And it oxidizes and smells really badly. There are these enzymatic products that you can buy that will clean up dog pee or cat pee, and they tend to have a class of enzymes in them called urease. They have ureases in them. So we tend to name the enzyme after the substrate that it works on. Okay. Enzymes usually, almost always, I I can't say always because I can think of a couple that don't. But enzymes' names usually end in ase. So lactase, urease, phosphatase, things like that, they end in ase. But an exception is that thing called lysozyme. That doesn't end in ase. That's an exception. Alrighty, so let's talk about how enzymes work quickly. Again, I know it's all review for you all, but I just want to remind you that enzymes work because they lower the energy of activation for a reaction. Let's see what that looks like here. If we took our generic chemical reaction, substance, come back here. Let's go with red right now. Substance A reacts with substance B to form products C and D in the presence of an enzyme, enzyme Q, I'm calling it here. We can assign an energy to the reactants. Well, I can't, but in the laboratory, scientists, chemists can do that. So if A plus B are at this energy level here, and the products are down here, C and D, we don't just draw a line like that. What the actual energy graph looks like, and let me get rid of that. What it actually looks like is, I'll put it in green, way up here. So this whole burst of energy needs to go into this chemical reaction to have it happen in the first place. That is called the energy of activation, or E sub A. And this is without an enzyme, whereas the red graph is with an enzyme. So this right here, it lowers the bump. The presence of an enzyme lowers the energy of activation. It lowers the amount of energy that's got to go into this chemical reaction just to even get it going. And the deal with calling enzymes catalysts, saying, oh, they speed up chemical reactions in the cell without getting used up or changed in any way, yeah. They don't just speed them up. If this enzyme isn't present, this is too much, this is too much of a hill to get over. A and B aren't going to react, and it's just not ever going to happen at all. So chemical reactions in the cell that require an enzyme, they don't just go slowly if that enzyme's not there. They're not going to happen at all. All right, so how do enzymes lower the energy of activation? Need to talk about that. They do it by binding to substrates at their active site. So here we go. We're talking about active sites. All right, so this is the active site. And here are the substrates. Substrates are just substrates are just reactants that bind to an enzyme at the active site. You've seen this all before. They bind. It's like a lock and key sort of thing. And here we, excuse me, I just burped. I just burped in lecture. Oh no. Here, let me get a drink of my tea. Anywho, this is the enzyme substrate complex. What the binding of a substrate to the active site does is that it is able to hold substrates in a position so that they can react with each other. Or in this case, it looks like this one's being broken apart and that the products are released. So maybe this is C and maybe this is D. Something like that. But I want to point out something to you, and that's in this, ball and stick thing down here. These ball models. All right, here we have our substrate right there. I'm going to erase that so that's out of the way. When a substrate binds to an enzyme, we kind of call it the induced fit model. So not really like a lock and key, but like a hand in a glove. That when the glove is off the hand, it has one shape and it's kind of flat, but when you pop your hand into it, it's more 3D. The shape of the glove has actually changed, especially if it's like one of those super stretchy gloves. It stretches to fit the substrate. It stretches to fit your hand. Now take a look at what's going on and how this looks down here. Down there. When the substrate is bound in here at the active site, can you all see that right there, how it's bound? Look down here again. I see like little 1, 2, 3 balls here, but I only see 1, 2 balls here. The deal is, and this is going to be important later on, so make sure you write this down, that when a substance binds to an enzyme, when a substance binds to a protein, it doesn't even have to be an enzyme. When a substance binds to a protein, it changes the shape of that protein. So let's write that down. Oh, it's the screenshot thing. I keep forgetting that I'm in screenshot mode there. All right, let's get a different color here. I like that green. When a substance binds to a protein, the shape of the protein can change. And it can change anywhere on that protein. Like, let's say there's an enzyme and here's its active site. Looks like that. I'm not going to color that in. It looks silly. And let's say it binds to substance A. This is the substrate. When we have a binding here, this ES complex, enzyme substrate complex, can change its shape, and now we have an extra site where maybe something else can bind to this protein or enzyme, if we're talking enzyme here. So keep that in mind, because that's actually how a lot of genes are regulated and turned on and off, and it's also how proteins are turned on and off too. Like sometimes a competitor, sometimes this is called, sometimes, what did I just do there? Go away. There we go. All right, I know that I put this on Do Not Disturb mode. Why am I getting things from TikTok telling me this stuff is up? Oh my gosh. Okay. Where was I with this? Oh, sometimes this is called an allosteric site. And we will see that later on in another chapter. Oh, is it chapter 10? When we talk about regulation of genes. But the point being that when stuff binds to proteins, the shape can change. The binding of this substrate here caused a change down here that opened up this site. And that kind of stuff happens all the time. All right, so we got enzymes. Go. Yeah, we got that. Oh, this is just to show you that sometimes there are cofactors that help create the active site on an enzyme. Cofactors and coenzymes will do that. So sometimes those are needed. Oops, why did I do that? Here we go. Yeah, they're just non-protein molecules. So they'll create an active site. Sometimes they help with the actual catalysis, like cutting a substrate in half or adding a new molecule to it. So cofactors and coenzymes, and we will see one example of that later on in this chapter. Oh, this is just to show you that enzymes have optimal temperatures, and they have an optimal pH as well. And you've seen graphs like this before in Bio 107. Here we go where we have enzyme activity on the y-axis and temperature over here. Enzyme activity will keep increasing until it gets to its optimal temperature, right around up here, and then it's going to drop off once it gets to a certain temperature. That's because the enzyme has been denatured. Likewise, enzymes get denatured at pH extremes. Enzyme activity is actually going to have a broader range of optimal pH than temperatures. Notice that the slope of the line over here is not as steep as it is over here. That's because heat is just going to denature the crap out of an enzyme. Denature the crap out of a protein, whereas a high pH alkaline won't be so good for it right away, but it doesn't drop off as quickly. It doesn't denature a protein as quickly as a very high temperature. So, and I just want to point out that the optimum temp is going to differ for different organisms. You know, this optimum temp for human enzymes is around 37 degrees C, and it's going to be the same for the mesophiles. E. coli likes it at 37 degrees C. Staph likes it maybe a little cooler than that. If we're talking some crazy archaea bacteria, they might have an optimum temperature down here. Alrighty. Just to refresh your memory about ATP, and this was already in one of our lectures, but just make sure that you know how ATP is used. This works better with that. It's got the adenosine end and 3 phosphates here. This initial phosphate is chopped off. That releases energy and loosens that phosphate group. Usually it actually gets attached to another molecule pretty rapidly. But we're left with a DP, adenosine diphosphate. That releases energy. But if we go the other way around to get the band back together, put these two back together to form ATP, again, energy has to go into that. And where this energy comes from is basically the subject of this entire chapter. It comes from the food you eat. It comes from the food we give to the microbes. So comes from breaking down or metabolizing foodstuffs, mainly carbs. Metabolism is, for the most part, mostly about the carbs, which is a good thing. I like to eat them. All right, so we got enzymes, we got ATP. Yeah, so this is just another graphic of this. That's what I showed you before. So ATP, you cut off, and you can't see me pointing at this if I don't do a screenshot. If we cut off this initial phosphate group that releases energy, we're left with a DP here. Energy needs to go into it to form ATP. Alrighty, so we got types of chemical reactions that we got to talk about here. Endergonic and exergonic. It sounds like endothermic and exothermic, but those actually release and use heat. Endergonic reactions are chemical reactions. Oh, I gotta use something you can see on here. There we go. Yellow shows up really nicely. Endergonic reactions require the input of energy to make them go. In the form of ATP or otherwise. Exergonic reactions are reactions that release energy. So an endergonic reaction, okay, exergonic reactions, we've got, let's say our A plus B, going to C plus D. And And we've got something that looks like that. That's actually an exergonic reaction, even though, because the overall, it goes down. This is not a good, no, this is not a good example here. I take back what I said there. Let's do this again. Let's say A and B are down here, and the products have energy up here. Can you see that that chemical reaction requires the input of energy to make it happen? That would be endergonic. Exergonic would be if it looked like this. A plus B are up here, and the energy of the products are way down here, and there we go. That's exer. And this is ender. And exergonic reactions can power endergonic reactions. So let's actually look back at ATP. Here we go. So which one of these is exergonic and releases energy, and which one is endergonic and requires the input of energy? So if this one, I should go back to red now, releases energy, this one is exergonic. It releases energy. So ATP being broken down and releasing energy, that is an exergonic reaction. And this one over here, ADP, getting its initial phosphate group back to form ATP, energy has to go into that. This is an endergonic reaction. And I'm pointing that out because exergonic reactions can be used to power endergonic reactions. And I want to talk about what those are as well. So to do that, we'll go back here. These are called coupled reactions. And again, I like to abbreviate reactions and save ourselves the writing. Let's say there is, let's go back to our generic chemical reaction again. Substance A reacts with substance B, even in the presence of an enzyme, to form substance C and D. So I'll write enzyme Q is required for that. You will sometime, well, I know you will see in your book something that looks like this. ATP becoming ADP. And they'll have this reaction arrow bouncing off the main reaction arrow. What this thing means here, what this means really is ATP with its 3 phosphate groups, losing its phosphate group, becoming ADP, and releasing energy. But check it out. If I erase that there, we can't put all this and write it there. What a hot mess that is. So we don't write all of this there. We write it this way. We just write, oops, we write ATP becoming ADP. And you just have to know that that releases energy. Now, what is the freaking point of all this? I will tell you what the point is. Energy is released right here in this arrow, right? But that released energy goes into the main arrow and makes this A plus B going to C plus D happen. So the thing is A plus B going to C plus D, is this ender or exergonic? Is A plus B going to C plus D releasing energy, or is it requiring the input of energy? And if you said it's requiring the input of energy, you are correct, and that would make it endergonic. So this one in green here, this is endergonic. And this one in yellow here, we know that's exergonic. It releases energy. And the energy that this releases is going directly in to power the main reaction, A plus B going to C plus D. These are called coupled reactions. Please make sure you get that straight, because I'll probably ask you that on the test, the next exam. But for example, let's say we've got a chemical reaction and it's A plus B in the presence of enzyme Q becomes product C plus D. But instead, it looks like this, ADP becoming ATP. You're like, whoa, the cell just made ATP. Isn't that cool? Isn't that the point of this? Well, yeah, but don't we know that this takes energy? This one here, this is an endergonic reaction. It requires energy to get ADP, its initial phosphate group, back to become ATP. Where's it getting that energy? Well, it's getting its energy from this one, from this A plus B going to C plus D. That must be releasing energy. So this one in blue must be exergonic. And the energy that it's releasing is going directly to power this one to make ATP. Coupled reactions. It's pairing an exergonic reaction with an endergonic reaction. So we got that there. All right, there is another type of reaction that we need to learn about. So that's endergonic and exergonic. There's another type we need to learn about, make sure we're all on the same page, about oxidation and reduction reactions. This probably sounds kind of familiar, but the definition in chemistry, and this is biological chemistry, is that an oxidation occurs if a molecule loses one or more electrons. And I'm just writing electrons with an E with a negative charge to it. If a molecule loses one or more electrons, it's been oxidized. It's an oxidation. A reduction is the opposite. It's when a molecule gains one or more electrons. Now, how are we going to remember this? Easy. We are going to remember this stupid little mnemonic, which is just so ridiculously stupid. It's that says that if you lose, no. Wait, what am I doing here? Stupid little mnemonic that is Leo, the lion, goes grrr. If you lose an electron, it's an oxidation. If a molecule gains an electron, it's a reduction. It's stupid, but Leo goes grr, lose an electron, it's an oxidation. Grr, gain an electron, it's a reduction. Sometimes people use oil rig. Oxidation is losing. Reduction is gaining. I know it seems weird that if something is reduced, it's gaining something. Doesn't that sound the opposite? Yeah. But that's how it is. All right, so let's see. Let's do an example. Let's look at an iron plus 2 ion. If an iron plus 2 ion loses 1 electron, it becomes an iron plus 3 ion, and this is an oxidation. Yeah. And this is the rust process here. And you learn about this like, okay, what has rust got to do with how cells make energy? Well, that's the problem. Because oxidation and reduction reactions were first, described by chemists who weren't looking inside the cell. They were looking in batteries and stuff like that. In the cell, things happen a little differently. In the cell, where an electron, let me phrase this differently. In the cell, when a molecule loses an electron, it usually also loses a proton. And you know that that thing is a proton. Talked about that last time, or 1 or 2 lectures ago. And also, when a molecule gains an electron, it usually also gains a proton. And this is just in the cell, the way this happens. Electrons just don't move around in the cell too often, except with some major exceptions. But when a molecule loses an electron in a cell, it usually, almost always, also loses a proton. So let's say we got this molecule here that loses 1 electron and a proton. What has it lost? What do these things here put together make? Well, remember why we call this thing a proton? Remember hey, come back, circle. A proton, that's just a hydrogen ion, right? What happens to this little hydrogen ion when we give it back its electron? It becomes a hydrogen atom, doesn't it? When we get these guys back together, when we get the proton back together with its electron, it makes a hydrogen atom. So we gotta, when we're talking about the cell, oxidation and reduction in the cell, we gotta go back and amend our definition. We need to say, how about purple? When a molecule loses 1 or more electrons or hydrogen atoms, it's an oxidation. When a molecule gains 1 or more electrons or hydrogen atoms, it has been reduced. So let's take a look at what on earth this actually means for cellular respiration. We'll check this out. C6H12, I'm color coding here, O6, plus 6O2, becoming 6, hey, don't do that to me, 6CO2 plus 6 water molecules. And I color-coded these things for a reason. All right, I'm looking over here at glucose, and I am 100% convinced that all of these carbons in glucose here get converted to CO2. Is glucose being oxidized or reduced? It's losing these hydrogen atoms, sure looks like it. And I'm looking at this going, when a molecule loses 1 or more electrons or hydrogen atoms, it is an oxidation. So, friends, this is an oxidation. And check out oxygen right here. Let's connect, hey. Keep forgetting that Notability does that. Is oxygen losing hydrogens or is it gaining hydrogens? It's gaining hydrogens. So is that an oxidation or a reduction? Leo goes grr, lose an electron or hydrogen, it's an oxidation. Grr, gain an electron or hydrogen, it's a reduction. There we go. So for cellular respiration, you need to know who's being oxidized and who's being reduced overall. And it's because in a cell, where a proton goes, an electron goes with it. So if a cell is losing an electron, it's going to lose a proton too. If a molecule in a cell is losing an electron, it is probably also losing a proton, which means it's losing a hydrogen atom. That's an oxidation. In the cell, if a molecule is gaining an electron, it is probably also gaining a proton, which means it's gaining a hydrogen atom. So that is enough for us to wrap our brains around today. We will talk about this in class while we Zoom, and I hope you have a great day.